tous à ce 18e webinaire conjoint entre le Chapitre Saint-Laurent et EcotoQ. Notre conférencière aujourd'hui est Sarah Wallace. Sarah est étudiante au doctorat, je devrais dire étudiante senior au doctorat, puisqu'elle est dans sa dernière année à l'INS. Et sa présentation est intitulée Identifying Gene Expression Biomarkers in Multiple Bird Species Exposed in Ovo to Diluted Bitumen. Sarah est étudiante dans le groupe de Valérie Langlois et est également co-dirigée par chaîne de SOLA, de Environnement et Changement Climatique Canada, si j'ai bien compris. Excellent. Donc, sans plus tarder, je vais laisser la parole à Sarah pour nous partager ses résultats.
OK, merci pour l'introduction, Claude. Okay. Well, so today I'm going to speak in English just because it's easier for me, mais je peux essayer de répondre aux questions en français à la fin de la présentation si vous voulez. So I'm really excited today to talk to you about one of my chapters of my thesis, identifying gene expression biomarkers in multiple bird species exposed in ovo to diluted bitumen. And as Claude said, I'm co-supervised by Valérie Langlois and Shane DeSola. And our collaborators for this project are Raphaël Lavoie, Gaël Trifot-Boucher, and Mason King. So to start off with, polycyclic aromatic compounds, or PACs for short, are widespread environmental contaminants with 2 or more fused benzene rings that are often present in complex mixtures. And with the infinite possibilities of alkylation, for example, adding a methyl group to any of the carbons, there's proposed to be over 100,000 congeners that could be in different proportions in these mixtures. These mixtures could be of pyrogenic origin, such as through industrial emissions or vehicle exhaust, or even naturally from forest fires, or even petrogenic in origin, such as in petrol or oil products. And in a review that we published in 2020, we looked at the levels of PACs detected in the tissue of wildlife in Canada. And maybe not surprisingly, most of the studies looked at fish and invertebrates. But most of the studies that we did look at that measured the levels of PACs within the tissue did not actually look at effects within the same study. And while vertebrates do have high metabolism of PACs, effects still can occur. And we identified the main ones as embryotoxicity, chorioallantoic, and oxidative stress. And although the mechanism of action are similar among species, the sensitivity does vary within and among taxa. And if we focus in on the petrogenic mixtures that could be transported by ship or rail, the one we hear most about are shipping by pipelines. So if we look at this map of the vast network of the oil pipelines in North America, it's no surprise that there's a high risk of an oil spill, and we do see these spills occur. And so we need to develop appropriate biomarkers or indicators of PAC exposure and potential effects. So one example of a petrogenic PAC mixture is diluted bitumen, or dilbit. It's a complex hydrocarbon mixture that starts off as this thick and viscous bitumen that's extracted from the oil sands. And through a series of dilutions with diluents or other condensates, it lowers the viscosity by adding low molecular weight PACs so that it can actually flow through the pipelines. But this actually adds in the 3 to 5 ringed alkylated pHs, which have been shown to be the most embryotoxic in fish. Recent studies have shown that dilbit is embryotoxic to fish and frogs. These are just 2 examples of studies here, where you can see the embryo on the top that's just exposed to clean water in the control condition look normal, but when they're exposed to dilbit, they have an array of different malformations. And we also saw this was true with the larval fish species in Roxanne's webinar about a month ago. We've seen that toxicity of dilbit to fish has shown through alterations in the transcriptome, or looking at all the expression of all the genes within the organism, as well as the proteome, or the levels of proteins. However, the mechanism of action of pack mixtures is not fully understood in birds. Only a few studies have actually shown the effects of dilbit exposure in birds. Birds could be first exposed to dilbit through ingestion, and this one study looked at adult zebrafinch that were orally exposed to Cold Lake blend, which is one of the most commonly transported dilbits in Canada. And they saw an increase in mortality, increase in EROD activity, which is an indicator of pack exposure, as well as decrease in active behaviors. Embryos, on the other hand, could be exposed to dilbit, for example, if their parents had oil on their feathers and then they went to sit on the eggs to incubate them. So this study looked at applying dilbit to the external surface of the eggshell, so essentially painting the dilbit on it. And they also used Cold Lake blend. They saw an increase in pack residue within the carcass itself, suggesting that these packs are going through the eggshell. But there were no differences in survival or the presence of deformities, and there's very limited evidence for aerobic capacity or growth. They did see increase in CYP1A gene expression, which is an indicator again of pack exposure, but they only saw it induced at the earliest and lowest application of the dilbit. So this still leaves some questions. Therefore, the objectives of my research are to assess the mechanism of action of dilbit toxicity through egg injection as a different exposure technique, and to identify biomarkers of pack exposure and effect in embryonic birds in general. So to do this, I had 3 species of interest. The first was the domestic chicken, which is a model species that breeds throughout the year, which was super helpful during the pandemic when doing fieldwork was not so easy. But it's also been used for in vivo exposure to single packs, so we can compare to those studies. And there are a lot of molecular tools available. I also looked at the double-crested cormorant, which is known to be a wild model species, as it's commonly used in toxicity studies. It does have a low to mid sensitivity to other contaminants, for example, compared to the chicken. And there are a lot of molecular tools available. I also looked at the Northern Gannet, which was an indicator species in the St. Lawrence Action Plan. And it was also one of the most affected species by the Deepwater Horizon oil spill, but molecular tools are limited. So I first collected fresh non-incubated eggs from relatively clean reference sites. And I think I have this slide a bit just to show off the fieldwork photos, but the cormorants, I collected the eggs from islands in Ontario, as well as Ile de la Maine and Ragueneau on the Côte Nord de Quebec. I got to go to Ile Bonaventure to collect the gannet eggs. And excitingly, I got to go 40 minutes north of INRS to the chicken hatchery there to collect the chicken eggs. In terms of the dilbits used, I looked at 2 different Canadian dilbit blends for this study. The first was Clearwater McMurray, which originates from the Athabasca oil sands, and Cold Lake blend, which originates from the Cold Lake oil sands. I then drilled a hole into the top of the egg in the air cell and injected dilutions of the dilbit into the air cell. This was on day 0, meaning that there was no incubation yet and there was no growth of, no embryonic growth at all. This is following the methods of the avian early life stage toxicity tests with a few modifications. And while it's more commonly done to paint the outside of the eggshells, Mason King, a PhD student at West, was already doing that with cormorant eggs and dilbit. So I wanted a different exposure technique where I could know for sure that the dilbit was getting into the egg and that the embryo was exposed to the dilbit. There's also the option of injecting it into the yolk sac, but The air cell is actually more practical, and you're less likely to get mortality just from like manipulating the egg itself for physical disruption. In terms of my experimental design, you can see I have the species on the left-hand side here. I had two controls. I had a non-injected control, which I did not manipulate at all, so no drilling of holes. I had a vehicle control, which I did drill a hole, but I injected the non-toxic corn oil, which is what I used to make the dilbit dilutions as well. Then either with clear water or cold lake blend dilbit, I injected a range of dilutions ranging from straight dilbit down to 1 in 10,000. So each egg here represents a treatment of about 15 eggs, but that did vary based on how many eggs I was able to collect. For example, chickens, I pretty much could get as many eggs as I wanted to, so I was able to compare the toxicity of clear water and cold lake blend within the same study. Versus in cormorants, I had to look at clear water and cold lake blend separately. And then in gannets, we only had enough to look at one dilution of cold lake blend. And so maybe you're thinking, well, this is not really an environmentally relevant exposure technique, you know, drilling a hole and then injecting the dilbit right into the egg. Maybe, but we tried to verify this by looking at the levels of packs within the injection solutions. So we measured that at CAQ, and it came back with a range of 200 to 300 nanograms of total packs per gram of egg. And this amount is actually similar to the external dosing studies where they painted the eggshell and then measured how much packs were in the carcass. So this suggests that maybe this exposure technique is relevant. But to look at and verify more of the environmental relevancy, I wanted to compare it to other studies that looked at packs within eggs in the wild. To do this, I distilled the results down to the US EPA priority 16 pHs, which many of the older studies just reported these pHs. So in both the clear water and the cold lake blend, I found a range of about 0.03 to about 7 nanograms of these 16 pHs per gram of egg. And these amounts were also found in eggs in the wild in Britain, as well as eggs from the oil sands area. So while it's, yes, these eggs could be exposed to other things, it still shows that these embryos in these experiments are exposed to pack levels that were within an environmentally relevant range. So hopefully I've convinced you, but if not, we can talk more later. So once these eggs were injected, I artificially incubated them until their liver was formed. I chose this stage because one, before the liver is formed in early incubation is a really sensitive time during development. And also for my previous chapter, my thesis, I did a microarray analysis where I looked at the transcriptome or all the genes, expression of all the genes within the embryo throughout development, but when they're not exposed, to look at when during a normal development, when are these genes or pathways being expressed. And I found that during this time when the liver is formed is a very time of big changes. So this happened in chickens. The liver was fully formed by 7 days. In cormorants, I sampled at 12 days. And in gannets, I sampled at 14 days. I could monitor the embryonic growth throughout incubation by candling or shining a bright light underneath the egg to look for the blood vessels. And then at the time of sampling, I sampled the liver as the main detoxifying organ, as well as the chorioallantoic membrane or CAM, which I'll explain in the next slide. But I preserved these tissues for RNA extraction and then did targeted gene expression by qPCR to look at specific genes within detoxification pathways to see if the expression of these genes increase or decrease with dilbit exposure compared to the controls. So what is the chorioallantoic membrane, or CAM? It's a highly vascularized membrane in the egg that has many functions during embryonic development. It actually touches the eggshell, and so it acts as the breathing organ, exchanging oxygen and carbon dioxide. It provides calcium to the embryo, and it also stores waste. It's known as an experimental model for angiogenesis, or blood vessels growth, as well as tumor grafting research. And it actually develops before the liver. It's known for physical protection of the embryo, because as an early embryo, it doesn't have, at points, it doesn't have a fully functional immune system. So the CAM acts as this physical protection. But what's also cool is that it also has potential for detoxification. So in chicken embryos exposed to PCBs, there was increased EROD in the CAM, which was relative to the amount of EROD induction that they saw in the liver. So as an additional objective to my project, I wanted to see whether the same genes expressed in the liver were also expressed in the CAM when exposed to dilbit or with PAC exposure. So to jump into the results, first we'll look at mortality. So on the x-axis, I have the two controls, the C being the non-injected control and the VC is the vehicle control, and then the range of dilutions of dilbit. This is mortality percent, so percent of embryos that were dead at sampling, which actually excludes any of them that were underdeveloped. So this is not fully like all the results, but this first just looking at the number of dead embryos. So here we see that chickens had the highest percent mortality, and in gannets, really there was none that were dead. And the cormorants were all over the place. In terms of malformations, we didn't actually see a pattern in the number of malformations with the dilbit dilution, but they were similar to ones that that have been seen in ovo exposure to single packs. For example, in the developed embryos here, you see cranial malformation or even underdeveloped bill. And these weren't really seen in the other species. So to go into the mechanism of action, here is a simplified version of the xenobiotic metabolism. When a PAC enters the cell, it binds to the aryl hydrocarbon receptor, AHR, which can dimerize with the aryl hydrocarbon nuclear translocator, or ARNT. This elicits a response of transcription or the expression of cytochrome P450, such as CYP1A, which hydroxylates the PAC to allow for conjugation with a water-soluble functional group, so that eventually it could be exported out of the cell, such as through these ABC transporters. But through this process, it can also produce reactive oxygen species, or ROS. And so also within the cell, you have ROS scavengers or other ways to detoxify the cell. So all of these genes and enzymes that are in these boxes are some of the most reliable biomarkers in fish that have been exposed to PACs. And in particular, CYP1A has been shown to be a great biomarker of PAC exposure and effect. And even in birds with exposure to single PACs and PAC mixtures, we see an increase in CYP1A. But the mechanism of action of exposure to PAC mixtures is not fully understood. And so in the study, I tested the expression of these 10 genes of interest. So to start off with, I will start with one of the isoforms of CYP1A in birds, CYP1A4. I know this is a lot of graphs, but bear with me. I have each of the species on one of the rows. On the x-axis, you have the treatments and the dilution of dilbit. And we're looking at the relative mRNA level of CYP1A4 in this instant. In either the liver in black or the CAM in red, the asterisks show a significant difference from the vehicle control, and the little T's show that there's significant difference between the two tissues. So here you can see there's a clear increase in CYP1A4 with increasing dose of dilbit. And especially in the chickens, there's a really high induction of CYP1A4 in the CAM, even compared to the liver. And this is with exposed to CLB. If you look at the embryos exposed to clear water, a similar pattern with an increase in CYP1A4 with the dilution. If we look at the CYP1A5, the second isoform, again, you're seeing this increase, maybe not as high as CYP1A4, but we see an increase in both the liver and the CAM, at least for in the chicken. And then with exposure to clear water, again, you're seeing this increase compared to the vehicle control. For another isoform, a cytochrome P450, CYP1B1, it's actually usually higher in extrahepatic tissues. So kind of makes sense that in the chickens here, we see a high induction of CYP1B1 in the CAM, but not really in the liver. But what's interesting too is that in the gannets, we actually see this slight decrease in CYP1B1 levels with exposure to CLB. And then with exposure to clear water, similar, where we see an increase in the CAM of chickens and not really significant, no significant changes in cormorants. So in summary, we really only saw dose responses with in the cytochrome P450 genes and not in any of the other genes tested. For both tissues, CYP1A mRNA response was at least the same or even higher in exposed embryos of chickens versus cormorants and then gannets, suggesting that the chickens have this high sensitivity in the CYP1A response in comparison to the gannets who have like a lower sensitivity. This is well known, I guess, that there is species variation in the magnitude and the concentration at which CYP1A is induced. And in general, we see that colonial water birds are relatively insensitive compared to chickens. But it was cool to see that this is also true with exposure to dilbit. In addition, CYP1 mRNA response was at least the same or even higher in the CAM versus the liver. And this induction happened at a lower concentration of PAX in the CAM versus the liver. So I wanted to investigate the mechanism of action within the CAM further. So to do this, I looked at one of the adverse outcome pathways that starts with AHR activation, and then through a series of molecular cellular changes, you have altered cardiovascular function, leading to increased early life stage mortality. And so if we focus in on the molecular part here, basically what it's showing is first that what I showed in the mechanism slide was that for the xenobiotic metabolism response, a PAC entering the cell will bind to the AHR, which can dimerize with the ARNT. And this elicits the response, say, of CYP1A being expressed. But ARNT can also bind with the hypoxia-inducible factor, HIF1A, which elicits the response of the hypoxic response pathway through genes such as the vascular endothelial factor, the VEGF. So I think it's interesting to see how ARNT is playing between these two different pathways. So to look at that, I looked at the expression of three genes, ARNT, HIF1A, and VEGF in the CAM only, but of the three species. I know this is a lot of graphs, but I will show you where to look. So if we zoom in on the hypoxia-inducible factor in cormorants, there was a significant decrease of HIF in all of the treatments for CLB exposure. But in gannets, we actually saw an increase in the expression of all of these genes. So if we map that back onto the AOP, or the adverse outcome pathway, with cormorants, we see this decrease in the hypoxia-inducible factor, which kind of makes sense with this pathway for strong AHR ligands that potentially could lead to early life stage mortality. But in gannets, it kind of seems to be the opposite, where we actually have an increase in HIF and BGF. So if we go back to thinking about ARNT being used for these two different pathways, I've added in also the results from CYP1A4. So potentially what I was thinking is that if we have an increase in HIF in the gannets, potentially ARNT is being used more for the hypoxic response pathway, leaving less to bind to AHR, and then maybe you have a lower induction of CYP1A. Versus in the cormorants with the decrease in HIF, it means there's a lot of ARNT left to bind to AHR and thus have a higher response in CYP1A. So to further investigate within this adverse outcome pathway, to look at whether there's any cardiovascular effects, I measured the heart rate, or the heartbeats per second. And in chicken embryos that are exposed to strong AHR agonists, they had an increased heart rate, but all packs are not strong AHR ligands. And we actually saw this decrease in heart rate in gannets exposed to CLB. This was in line with zebrafinch that had their eggshells painted with Deepwater Horizon oil, who also had a decreased heart rate. So we can add that to the adverse outcome pathway. And it seems that with gannets, even though they may have this opposite response of the increased hypoxia pathway, they're the ones that actually showed a decrease in their heart rate or alteration to the cardiovascular function, versus the cormorants that we saw this decrease, but we didn't see any changes in the heart rate. So I guess it's not a surprise again that species have different sensitivity. And with CYP1A in particular, to summarize, chickens had a very high induction of CYP1A versus the cormorants that had somewhat high. And then in gannets, really, there was not much of an induction of CYP1A, but we did see effects in the mechanism of action within the CAM. So to try to think about maybe why this might be, first idea is, well, maybe it could be because of the AHR binding sequence, for example, where chickens are the most sensitive subtype versus cormorants and potentially gannets that have the least sensitive type. But work coming from Jessica Head's lab at McGill show that, the relationship between the AHR subtype and species sensitivity does not really seem to extend to pHs like it does with other strong AHR ligands. Maybe it could be the total duration of the natural incubation period. For example, chickens of 21 days versus gannets of 45 days. And not just for the full incubation period, but also their developmental timing. Because for example, when I sampled the embryos when their liver was formed, it could be that their CAM is actually at different stages of development. So in mallard ducks that have a similar incubation period to cormorants, when their liver was formed, their CAM was not fully developed, versus chickens, when their liver was developed, they had fully vascularized CAM. So maybe this difference in developmental timing of the CAM could be changing why we see, or show why we see differences in their response. Maybe it could be the number of eggs that they lay. For example, chickens that I took the eggs from are laying eggs all the time. Cormorants lay 3 to 5 eggs per breeding season, and gannets essentially only have 1 successful egg per breeding season usually. So potentially there's differences in their development or in their maternal input as well. So in summary, we see and we know that pack mixtures are complex. The 2 dilbit blends that I looked at do have different proportions of their packs within the mixtures, and we do see slightly different results. See that the CAM plays a role in detoxification, where the same or even more pronounced biomarkers are seen in the CAM versus the liver. And what I'm interested in is whether the CAM can help to detoxify the egg or help protect the embryo before even the liver is formed. And so I think it should be considered as a tissue to look at for exposure and effect. We see that species respond differently to the dilbit exposure through egg injection. And also that CYP1 genes are induced in early developing bird embryos, and so they are good biomarkers for PAC exposure for multiple bird species. And so I think I still have some time, so I would love to tell you about my next project quickly. So for my next chapter, I'm looking at PAC exposure in bird blood. So levels of PACs detected in blood of birds were associated with different effects. And this is an in vivo exposure. We saw a decrease in hematocrit or an increase in oxidative damage. There's also effects seen in red blood cells, particular, when exposed to PACs in vitro. So this is increase in oxidative damage, increased DNA damage, and increase in proteins such as the transport protein, P-glycoprotein. And what's cool is that red blood cells are nucleated in birds and fish, and they're responsive to xenobiotic exposure by changes in their gene expression. So my question is, do bird red blood cells respond to PAC exposure in vitro? More specifically, my objectives are to assess the applicability of using red blood cells as an in vitro bioassay for PAC exposure. So essentially the ideas would be to reduce the number of eggs I have to collect or the number of embryos used. So I want to determine at what PAC concentration and at what duration of exposure any sensitive biomarkers in red blood cells may respond to this PAC exposure. So I've isolated red blood cells from chicken blood, and I exposed in vitro these red blood cells to a range of concentration of benzocaifluorantine, which is was shown to be one of the most potent pHs to bird embryos. And in addition to the negative controls and positive control, I sampled the red blood cells at differing times of exposure. And so at this point, I've done a couple of pilot studies to test that the methods do work. And soon I'll be doing the full exposure, which I'll sample the red blood cells for non-targeted omics. So instead of just looking at 10 genes, I will look at the whole transcriptome. We'll look at all the proteins within red blood cells and all of the metabolites using metabolomics as well. So I'm really excited to see at which level, if any, the red blood cells will respond to PAC exposure, but you'll have to stay tuned to see what the results are. So with that, I would like to thank my funding sources for my salary as well as the projects. Thank you for access to the sites and for the collaborators for our field help, and then our hydrocarbon analysis that was performed at CAQ. Thank you to the members of the Langlois Lab for helping me and support through all of the difficult exposures. And I'd be happy to take any questions. And because I am in my last year of my PhD, I am starting to think about what's next and if there's any postdoc opportunities. So if you have any ideas, I'd be happy to chat as well. So thanks for listening.
Thank you, Sarah. We are now in the question period. Est-ce que nous avons des questions pour Sarah? Vous pouvez utiliser le chat, lever la main ou tout simplement activer votre micro pour poser une question à Sarah. Sinon, on a... Oui.
J'ai plusieurs questions. Well, I guess I should speak English.
Okay.
Although your French is really good now, I'm impressed.
My one line, yeah.
Yeah, good job. Yeah, I have a million questions and I'm looking forward to talking to you more, but just to start, do you think that the metabolites of the pHs are in the CAM?
Yes, I think so, because metabolites from metabolism from CYP1A, if it's happening within the can, that I would assume that those metabolites would be there as well. And because the blood is going through the cam, I assume then they would travel to the embryo as well.
Yeah. Yeah, the reason I ask, I guess, is it's really interesting, like what you've exposed them to is obviously a complex mixture of all different kinds of AHR ligands.
Yeah.
And you did see like the chicken being more sensitive than the cormorant, which is unlike what we've seen for, as you pointed out, for individual pHs. Right. So I'm wondering, like, you know, like how we can figure out what happens in these complex mixtures, because the metabolite, like they're all being metabolized differently in different tissues, and that certainly must contribute to the toxicity, right? It's like not as simple as just AHR plus one chemical equals
for sure
toxicity.
And whether some of the pHs are like stuck in the cam and that's where they're being metabolized versus getting into the liver of the embryo too. Mm-hmm. Might be interesting to measure what pHs are in both of those tissues, and also, yeah, even the metabolites too. But as you know, it's hard to measure everything.
Yeah, totally. But that might answer some questions about like why the CYP1A is induced more in the cam even than in the liver, which was really kind of interesting and surprising. And yeah,
that's cool to think about. Yeah.
Thanks.
Can I just add something there? Can I ask something too?
Yeah.
It looked like your relative expression of CYP1A 4 and 5 was relatively greater in comparison between the Cold Lake blend and your clear water.
Yep.
For your chicken as well. So it seems like, I don't know if that's a good comparison between the two, but it seems like you have differences between oil type. So there's different compounds in that oil as well, right?
For sure, exactly. So there's like, if you compare the concentration of different individual pHs within the mixtures, there are the higher proportion of the lower molecular weights within CLB versus clear water. I think clear water was more weathered than the CLB too. So that could be why there's a higher response of CYP1A in general for both of those CYP1A 4 and 5 with exposure to CLB versus clear water.
Yeah. So kind of similar. It was just touched on. You have differences in like the chemical profile of your oils, which kind of gives a different response in your CYP1A expression. I don't know. It just seems interesting.
And I'll show you that quickly. The secret slide. I don't know if you can see the it with the small text, but basically the orange is CLB. So you do have like a bit of a higher concentration in CLB for these lower molecular weights versus the higher ones. But in general, they are pretty similar, I would say. I think that's why it's important to measure, if you're looking at a mixture, you know, to understand what is the actual mixture and then how does it differ between mixtures.
Yeah.
I think there's a question in the chat. Should I answer that? She's wondering if the chemical gets transferred from the cam to the liver once formed. That's a really good question. I think that's similar to what Jessica Head was asking with if it's being metabolized in the cam, then what is moving into the liver? The liver is the main detoxifying organ, so I would assume that at some point, you know, these are being transferred there to be dealt with, but I think it's cool that the detoxification process can happen in the cam as well. And that's what I'm interested to know too, like in the timing of the development. If embryos are exposed really early on, can the cam help to detoxify and then you don't see as many malformations even before the liver is formed?
That would kind of make sense, I think, based on some data we've been seeing lately where these are just single pHs, like they disappear so quickly. Like the liver forms and they're gone. So maybe they were actually, the cam was working away on them beforehand. That makes sense.
Right.
Yeah.
Mhm.
And even for what you saw, like you saw pretty low fold inductions for some of them, for some of the species, right? Like 2 to 3 fold induction, which is like you'd expect with that mixture of pHs that you gave them, you'd expect it to be a bit higher maybe.
Right. Exactly. And so that's a good point, kind of answers what Jonathan's asking in the chat about what day I performed the qPCR assays on. So I did them when I sampled when their liver was first formed. So it was day 7 in chickens and day 12 in cormorants and day 14 of the gannets. And that's because of the high metabolism. What Jessica's saying is that as soon as they're exposed, basically they're gone. And so my idea was I still wanted to see, are they being exposed at that time? And so that's why I sampled them and did the qPCR on the tissues right as they were formed.
Any other questions for Sarah?
Yeah, there's one in the chat from the cormorant itself.
Yeah, who is that?
Actually, that's an old name for cormorants. Now it goes by nanopterums, so he's wrong. Now I know who it is, but always asking me the hardest questions. Okay, most studies on pHs focus on CYP1A and 1B, but other CYPs like 1C1, 1C2, 1D1 have shown ability to metabolize pHs. Do we know what the relative inducibility of these other CYPs are in cam and liver? Yeah, I'm not sure actually. I know that we always look at 1A and 1B because those are the highest and induced one, but I'm not sure if there's studies with pHs specifically for the other CYPs. Jessica, do you know?
Can't think of anything off the top of my head.
Yeah.
John, do you know?
I think there were in
Okay.
PCBs, and I think it's unclear as to whether all birds have CYP1C1s or CYP1Cs. I think chicken does, but for instance, I think quail doesn't have basal expression, but I'm not sure.
The other, with John speaking now, one of the other interesting things would be to try your dilbit blends in the LRG assay to see how they bind to the HR.
Yeah, that'd be really cool actually.
It might kind of, I mean, it's just really, yeah, I'm still trying to piece out like the evidence you have for differences in species sensitivity. It doesn't really line up with other stuff we've seen, which is interesting. So
right, just try to figure out why that might be. Yeah.
Yeah, it's like a clue.
John was asking, for mixtures, could competitive binding to the HR ligand binding domain affect relative gene expression? For sure, I think when you have the mixture and you have all these different packs, some bind more strongly than others, some act differently, don't even bind to the AHR and can affect other pathways. So I think that it's not a summation, like it's not because you have 2 packs there, you're going to have the induction of CYP1A twice as much as just one. So I think that's why it's difficult to make a direct link as well. It'll be fun to chat later with you guys too about it.
Another question in the chat.
Oh yeah. The exposure of the eggs was in another form, more normal. Do you think that we'll find different results? It's a good question. My idea again was to do this injection into the egg because I wanted to see sort of like a worst case scenario so that the embryo is actually being exposed. So it'd be interesting, now we know that these genes are changing in the cam and the liver with this type of exposure. So it'd be cool to try those genes within other exposure techniques, such as like applying it on the eggshell. And so maybe if the concentration of the packs is not as high, then maybe you would not see as high of an induction. Okay, sorry, another question. Would you consider repeating this experiment until hatching to measure survival and observe deformities, or would it not be as relevant? Well, there are lots of studies that do look at all the way to hatching and at different time points along the way. I think, like as we talked about, that packs are highly metabolized. And so I think if we were to see any effects, we would see them at this point. But of course, if you were to leave them to develop to hatching, you might recognize and see other problems or issues or malformations too. But they would have probably metabolized the packs already if you don't re-expose them. So yeah. The cormorant is asking, are constituent forms of SIP important for metabolism of pHs, or is it only inducible forms that are relevant? Well, I think in terms of looking at pack exposure, the ones that are induced are the important ones to look at. In terms of looking throughout the whole pathway, the other ones could be important too, because you have downstream effects that may be changing as well. So I'm not sure. Thanks, Jessica, about writing about the developmental sensitivity windows.
Well, it was nice to hear from the cormorant itself. Any other questions from humans or birds?
I'm going to use that trick next time.
All right, well, thank you very much, Sarah, and thank you everyone for your this lively discussion after the presentation. Je crois que cela conclut notre séminaire EcotoQ Chapitre Saint-Laurent pour aujourd'hui. Merci à tous et à toutes pour votre participation.
Oui, merci.